Hello everyone, and welcome to our online lecture series for Bio 2. Today we're going to continue our exploration of everything about the central dogma of biology, the idea that one gene codes for one protein. Well, last time we talked about the process of replication, where you begin with a single DNA molecule that's double-stranded, and will end with two DNA molecules that are each double-stranded. The basic idea of replication is that you will take each side, each of the individual strands of the original molecule, make sure they're untangled with topoisomerase, unzip them with helicase, keep each safe and protected with single-stranded binding proteins, and then copy them from each of these template strands, the original strand providing a template to copy our next new or newly synthesized strand of DNA. The components of that synthesis begin with primase, an enzyme that binds or anneals an RNA primer to the start location, where your RNA polymerase 3, the enzyme that's going to be doing all of the copying and all of the building, will attaches at that primer. But, as we spent a copious amount of time discussing last time, RNA polymerase can only build in a single direction, from 5' to 3'. This causes a situation where one strand is the leading strand, because DNA polymerase 3 is building in the direction that it should be, 5 to 3, and a lagging strand with our separate Okazaki fragments. And this is caused by the fact that the directionality of this strand would usually be going 3' to 5' to be opposite what the other side is, but we can only build 5' to 3'. So that means DNA polymerase must bind, build, hop off, bind, build, pop off, bind, build, and pop off in segments, or Okazaki fragments. Once DNA polymerase 3 has extended and added the new bases, DNA polymerase 1 will come through and replace the RNA primers. After all, RNA is not stable for long periods of time, as we'll talk about more today. And then, to wrap things up, we had DNA ligase connecting our separate Okazaki fragments to result in two newly synthesized strands of DNA. It also recommended not forgetting mentioning the sliding clamps, these proteins that help keep DNA polymerase attached, because if you remember at the end of last lecture, we talked about how fast DNA polymerase 3 moves, up to 1,000 bases a second in some of our prokaryotes, and the need for a sliding clamp, or in eukaryotes, the pCNA, to keep the polymerase on task and on target. Hopefully this makes you appreciate how much of a team effort DNA replication is, and also appreciate how much we understand about it. To extend our understanding of the central dogma of biology, we need to move on from this replication of DNA to more DNA, and instead explore the processes that take us from DNA to RNA, and from RNA to amino acids. And when a bunch of amino acids are joined together, we call them a polypeptide. That's a fancy way of saying a protein. We call them a polypeptide because they have many peptide bonds. The process of going from DNA to RNA, because we are staying in the language of NAs, nucleic acids, but we are changing the dialect from deoxyribose to ribose. That change of a dialect is a transcription process, where we're not changing the language, but we are kind of changing some of the words. When we go from the language of nucleic acids to amino acids, we have to do translation. And so today's lecture is going to be covering all of the steps of transcribing this segment of DNA into RNA and then translating that RNA product into a chain of amino acids. So, we definitely have to begin with a discussion of what exactly RNA is. So, RNA, much like DNA, is comprised of bases. But the bases are slightly different. Rather than the canonical A, T, G, C that we see in DNA, RNA uses adenine, uracil, guanine, and cytosine. One different base, uracil. Another feature of RNA, in contrast to DNA, is that it's often temporary. It's unstable over long periods of time, and it's often single-stranded. The unique component of RNA that really distinguishes it from DNA is its ability to form three-dimensional structures on its own. This allows RNA to have some enzymatic properties, meaning that RNAs can catalyze reactions on their own. For example, this is an RNA molecule that has formed a hairpin loop on itself. So we'll follow the RNA backbone here. I'll grab a little thicker marker here. You see that it starts here, up, over, around, like a roller coaster, to form a hairpin loop on itself. DNA does not do this. DNA will generally form a long double helix with another DNA molecule. RNAs, in part because of that property of uracil, which looks like this, are able to form these three-dimensional structures. So what exactly is uracil, and how does it compare to the letter it replaces? You'll note that the letter it replaces is thymine, the T. Uracil is actually a demethylated thymine, meaning that it has one less methyl group in it. You can also form uracil when cytosine is deaminated. Meaning that you can get uracil from instances where cytosine becomes damaged. Now, I don't expect you to know what demethylation or deamination are, but rather be able to identify that uracil is not as stable as thymine, because you can make it from different ways of damaging different bases. The idea of why this is so important, the long-term stability of the molecule is so important, is that because RNA is generally short-lived, there's been no selective pressure to replace uracil in these molecules. So we see uracil persist in short-lived RNAs, but be replaced in long-lasting DNAs. Pretty crazy, huh? But there isn't just one type of RNA. Obviously, by the writing on the bottom of this page, you can see that there are 3 different types of RNAs. So let's take a look at these 3 different types now. So the 3 different types of RNAs are messenger RNA, or mRNA, named appropriately because it carries messages. Transfer RNA, or tRNA, because it carries amino acids. And ribosomal RNA, because it is the structural piece of a ribosome. Let's go over them one at a time here to highlight some of their differences, because they're all made in that same transcription process, but what they do is very different, though it does intersect in the process of translation. Each of these RNA components, mRNA, tRNA, and rRNA, will reunite to complete the process of translation. Messenger RNA is oftentimes our single-stranded RNA, our typical RNA that we're thinking about when we think our single-stranded RNAs. It maintains a straight structure frequently because the idea is that it needs to be read. It is organized much in the same way that we see our DNA, meaning that it's just a string of a whole bunch of nucleotides. But underlying this code, we know that the mRNA can be broken up into codons, which are triplets of nucleotides. And these codons are said to be collinear because 3 nucleotides will always code for a single amino acid. So 3 letters of mRNA will result in the production of 1 amino acid. In order to do this translation, we have the codon table, as you see here. All possible iterations of all of our nucleotides of RNA, U for uracil, C for cytosine, A for adenine, and G for guanine. And because it's in 3 letters, we see the first letter on the left, the second letter on the top, and then the third letter dividing each of our boxes into 4 different parts. So for example, if you had first letter U, second letter U, third letter U, you would code for the amino acid PHE, or phenylalanine. Now this is going to be important when we revisit mutations, so I do want to introduce you to it here, but we'll spend some more time working on this later. For now, let's go ahead and practice doing this process of transcribing and translating our mRNA, given that original sequence of DNA that I gave you just below DNA replication, for consistency's sake. If this is our DNA, or this is our coding sequence, CDS as it's abbreviated, we know that this is what our mRNA is ultimately going to look like. In order to make an mRNA from this, we're going to have to read the opposite side. So therefore, let's go ahead and complement our DNA here. This means that we're looking at T pairing with A pairing with T, C pairing with G, G pairing with C, T pairing with A, G with a C, T with an A with a T, C with a G, G with a C, T with an A, C with a G. Voila! You've complemented your first strand of DNA. And no, that does not mean you're looking quite nice today. I like your jacket, DNA. It is instead adding the base that you would expect to see on the opposite side. DNA complementation. From this non-coding side of DNA, we then can mimic the process of RNA polymerase, the enzyme that's going to go ahead and do our transcription in order to make our RNA. From this strand of DNA, the resulting strand of RNA would look like this. A U G C A C A U G C A G. And it would still be one strand, with each nucleotide being separate. You'd see no delineation. But you'd know that underlying this, you would be able to group your RNAs into triplet codons. The AUG being read together, the CAC, the AUG, and the CAG. So let's go ahead and translate this sequence, starting with our AUG. A is the first letter, U as the second letter, G as the third letter, meaning that we make met, or methionine. Oh, I'll just keep it short as met. That's the amino acid. Coincidentally, you'll notice that methionine was highlighted in red here, because AUG, or met, is the universal start codon. That means that every single protein on planet Earth is going to start with AUG. Whether you're performing transcription in bacteria, or transcription in humans, or transcription in a dog that you occasionally hear barking in the background, like maybe a few minutes ago. All transcription, or all translation of proteins, begins with our AUG start codon. Next up, let's figure out what CAC codes for. We'll begin with first letter C, second letter A, third letter C. We see that it codes for the amino acid HIS, or histidine. Not important that you know what histidine does. That's for your next molecular biochemistry class. But important that you are able to figure out that CAC translates into HIS. Next up, we again encounter AUG, and we know that that codes for methionine, MET. And then last but not least, we have a similar strand, beginning with a C, advancing to an A, and our fourth letter being a G. CAG codes for glycine, not histidine. So you will notice that in many cases, the code is redundant, with the third letter not mattering for some amino acids, like leucine and valine and alanine, threonine or proline or serine. But in some cases, it makes a big difference. You might get TYR, or you might get the three different stop codons, the end signals for when the protein construction needs to end. And we'll go in greater detail as to what exactly a stop actually is in a little bit, but know that there are one singular start and three separate stops. I want you I will not expect you to memorize the stops, but just for ease of life, the start is always going to be AUG. So you can commit that one to memory. In DNA, it's ATG, and in RNA, it's AUG. So that was a lot, kind of quickly, about mRNAs, but we'll be revisiting a lot of topics to do with translation later this lecture and over the next few lectures. We can move on temporarily to tRNAs, or transfer RNAs. Unlike our single-stranded, non-three-dimensionally structured mRNAs, tRNAs end up having a pretty cool three-dimensional structure. In fact, we can describe it as a clover leaf. So it's got this kind of three-pronged endpoint with a top. You'll notice that it starts and ends at that top. We also know that this top piece, the acceptor stem, is where you will have an amino acid binding. So each tRNA carries a specific amino acid. The amino acid that it carries is determined by whatever nucleotides are in the anticodon position. So you will have that anticodon position binding to the sequence of the mRNA. So if you had AUG on the mRNA, you would bind to it UAC, binding to AUG, and at the top of the tRNA that carries AUC in the anticodon loop here at the bottom, you would find the amino acid MET attached. And that's the basic idea of how this works. You have each tRNA with all of our different variable codons carrying different amino acids that will then be carried over to our ribosomes to perform the process of translation. So, what are our ribosomes? Well, in large part, they're structured by ribosomal RNA, which is our major structural component, structural piece of ribosomes. In fact, about 60% by weight of a ribosome is our RNA, and 40% of the ribosome is proteins. So in this picture here, you see the rRNA in the tan color and the proteins in the blue color. And this is a still of a three-dimensional spinning image that you can find on Wikipedia of the small subunit of a ribosome. Ribosomes themselves are made up of a large subunit that I will very eloquently draw as the top half of a hamburger bun, and a small subunit. Very eloquently named because it is smaller than the large subunit. The small subunit is going to be interacting with our mRNA in large part, and the large subunit is going to be interacting with our tRNAs. Perhaps slightly oversimplified, but it allows us to better keep track of things when we move into translation. So with our three different RNAs covered, we now can actually get to that process that we began with by talking about transcription. So, transcription itself is broken down into three primary steps: an initiation step to get things beginning, an elongation step where a polymerase is copying bases and building the RNA molecule, and then some sort of termination step, an ending signal. And this three-step process of initiation, elongation, and termination will also be repeated in translation. But the circumstances will be slightly different. The major guide of transcription is one giant molecule, RNA polymerase. Now know that in eukaryotes there are different versions of RNA polymerases that do slightly different things, but we can kind of gloss over some of those details for now and focus on the fact that there is just one major enzyme guiding transcription. Compare that to the couple of enzymes that are involved in replication. Now, RNA polymerase is going to be doing all of the initiating, elongating, and then waiting for some sort of termination signal. So there are ways to communicate with it. Generally speaking, initiation will begin when RNA polymerase binds at the promoter sequence. In my kind of corny, comedic way, I always think of a promoter as the promoter to a club, a nightclub, a dance club. And so you have this sequence that's in the DNA sequence, so a component of this, but because of the chemistry of that sequence, it does a good job recruiting RNA polymerase. Some of those sequences that do a good job recruiting RNA polymerase, some of our common eukaryotic promoters, are things like a TATA box. Very cleverly named because it is a repeat of the nucleotides TA, TAA, TA, TAA. As it turns out, that's some of the pattern, the motif that RNA polymerase is looking for when trying to begin initiation. We also have some other different promoters, like the GC-rich box. And we literally call these boxes because when we wrote them out the first time and we're studying them, we put a box around them. And so, TATA box was born, and GC-rich box was born, and the CAT box, CAAT, CAAT, and the octamer box. A little bit more complicated, but it generally has to do with repeating 8 different bases. These promoter sequences allow for RNA polymerase to bind. Once RNA polymerase is bound, we then go to our elongation step. And speaking about elongation in eukaryotes specifically, we require a heterodimeric enzyme or a large enzyme called FACT. Again, in that cheesy comedy way, it is a fact that FACT is needed for elongation. Because FACT stands for facilitates chromatin transcription. Yeah, that's right. We got clever by taking first 2 letters, first letter, and turning it into the name of this giant enzyme. Facilitates chromatin transcription. It does so by working with RNA polymerase II in eukaryotes, like us, to help make sure that the polymerase is going to be moving. So while RNA polymerase is necessary and sufficient for performing elongation in prokaryotes, you have FACT helping it along in eukaryotes. FACT itself is actually cleaning up the upstream portions of our DNA. Because you might remember that DNA or chromatin is organized into nucleosomes, which themselves are 8 histone structures. And this allows the DNA to kind of be held together like beads on a string and preventing it from being super tangled up. But those nucleosomes and those histones are actually getting in the way when you're trying to perform elongation. So FACT is a histone chaperone. So you can think of it as a molecule that's going just ahead of where RNA polymerase II is and clearing the histones out of the way. Like, scoot, 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 scoot. We need to make our way through here. Now, this process will continue on up until you get some sort of termination signal. Now, the termination signal, whether you are a prokaryote or a eukaryote, is actually quite different. In prokaryotes, we have what is called a row-dependent signal. So there are some other intracellular communicators that actually tell transcription to end. In eukaryotes, conceptually, it's a bit simpler. Transcription will continue along, doop, 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 and continue and continue and continue up until the polymerase kind of slides off. Now, oftentimes this actually comes up against the long sequence of repetitive elements, so something that the polymerase finds kind of slippery or chemically slippery, but the RNA polymerase will just slide off, and voila! By tripping up, termination is reached. You have created your RNA molecule. But that's not quite the end of the story. The RNA molecule is not yet ready to go off and be translated. If you remember from your previous biology courses, the production of proteins happens at ribosomes. And ribosomes are either attached to the endoplasmic reticulum, the rough ER, rough because they have ribosomes attached, or floating around in the cytoplasm. Transcription is happening within the nucleus. The nucleus and the cytoplasm are not the exact same place. So we have to be able to get our mRNAs from the nucleus into the cytoplasm without them being immediately destroyed or being broken down. Remember, RNA is very temporary. There are enzymes on your fingertips that are meant to break down RNAs the moment they interact with them. And the same is true for a lot of your cytoplasm. It's a big, dangerous world for RNAs. So we need to do some RNA processing in order to make sure that they get safely to the correct location. So your primary RNA transcript will look something like this. It has a header sequence and some tail sequence, and then it has a series of exons that will stay because they are expressed in the protein, and introns that get removed. We're going to need to delete these. And we have exons and introns and exons. The introns need to be removed by RNA processing or RNA splicing. We won't talk too much about the spliceosome. Again, a giant complex that's really actually pretty cool and works alongside RNA polymerase. But RNA splicing is actually often happening concurrently with RNA transcription. So as you are making this primary transcript, you are continuing to make the tail end while the front end is being spliced. It's kind of all happening at the same time. The pictures of this are pretty cool. The spliced RNA will have a couple of additional features. Up front, it's going to have a 5' cap. And it's called a 5' cap because, guess what? That was the 5' end of your DNA. So again, it just corresponds to the same side as the 5' end. You'll also notice, what is the directionality of RNA polymerase? Well, it goes from your 5' to your 3' in terms of building. So that means that your RNA polymerase operates in the same direction as your DNA polymerase. 5' to 3'. Granted, there's a lot of things that are complicated and confusing in all of these processes, but one thing I have been always very thankful for is that this directionality did not change between the two polymerases. Whew. Your 5' cap is going to be the sequence on the front end of your RNA that is going to help find the right location and help you find the small subunit of a ribosome. So it's a protective piece. It's finding the correct location for where you need to be translated. So either moving you to the rough ER or the cytoplasm, and helping you actually bind to the small subunit of the ribosome. After your 5' cap, you have something called the 5' untranslated region, meaning that this component of the sequence is not going to end up in the protein. Oftentimes, this is because you do not have a start codon here. The first exon will begin where you have the first start, the first MET or AUG codon present in the spliced RNA. So exon 1 will begin with a start codon. Everything upstream of it is that 5' UTR for short, untranslated region, and it also can help with this locating and finding of the small subunit of the ribosome. After exon 1, you'll notice that intron 1 has been removed and exon 2 is adjacent to exon 1. After exon 2, you'll see that exon 3 has been placed adjacent to exon 2, removing that second intron. After exon 3, which will end in the stop codon, of which there are 3 different types, you will find a 3' UTR, or the tail end UTR. And after the 3' UTR, you will literally find a poly A tail. In order for this spliced mRNA to not be immediately destroyed and shake off some of the things that might be trying to destroy it, a poly A tail, or a long sequence of A's, is added to the end of your processed mRNA. Now, just as an illustration that I might be glossing over some of the underlying details here, I want to talk explicitly about that poly A tail for a second. So, I don't like to sugarcoat over things, but there's just kind of a lot to understand as to what each of these introns and exons and UTRs and 5' caps do, but just know that they're not all just there. They have purposes. What we find with the poly A tail is that over time, the poly A tail will shorten. And when it is short enough, the RNA is actually degraded. You break down the RNA when the tail has become short enough. So in this way, the length of the tail is kind of a timer for how long you're going to be using this mRNA to make proteins. And in this way, the structure of your spliced RNA can actually be informative to overall gene expression of the protein that you're trying to make. I think that's a pretty neat and fairly clear example of how all of this splicing has downstream effects. Everything has a purpose the more you study it. And in this way, I just want y'all to be aware of the fact that the poly A tail kind of serves as a timer for mRNA expression. Once you've got your finalized transcript, we then can move on to the process of translation, which, as I've mentioned before, begins with initiation, continues with elongation, and ends with termination. Here's where I'll actually be drawing this process, because it's an iterative kind of chain reaction where it helps to see things as they're being added. So you will begin with your, let's go ahead and grab it, it was our pink sequence, your processed mRNA. Now, over here will be your 5' end, over here will be your 3' end, and so somewhere around here you will have, your start. Your small subunit of your ribosome will bind to the mRNA. Once the small subunit binds, it then goes ahead and recruits a large subunit. Your large subunit is defined by 3 sites: the E, the P, and the A, or the exit site, the polypeptide site, and the amino site. This structure begins with the start codon at the A site. In the A site, you are going to find a tRNA. I think I had tRNA in green, actually. That is carrying some sort of amino acid, like MET, that will have its anticodon sequence binding to the codon present in your start. AUG on the mRNA will bind to UAC on the tRNA. This tRNA will float into the A site. It will bind here to the codon, and once the binding is complete, this whole structure will tick over to the P site. Now, at the same time that the tRNA is moving over to the P site, you have the mRNA and the small subunit ticking over as well. But the small subunit will tick back. So that way you've literally just slid the mRNA forward. Now this frees up the A site. At the A site, you will then get another codon that will wait for a tRNA to come in. A tRNA that carries a different amino acid. Like, I shouldn't change color here. Well, it might make more sense. Like glycine, GLN. This GLN, carried by this tRNA, will bind to its anticodon at the A site. And then what happens is a little bit tricky, and my drawing ability here kind of fails. So what actually happens is that the methionine will get bumped up. So it gets ticked up, not the whole hat here. This tRNA gets removed, so it leaves through the E site, and the glycine carrying tRNA then moves through to the P site. So again, everything shifts over, the small subunit clicks back, and now your MET will actually be sticking on top of your glycine. And in this way, what it ends up looking like is that you always have a tRNA here, an amino acid will be stitched on top of the tRNA, and the previous amino acid will be stitched onto the next one. And when a new tRNA moves in to the A site and then the P site and clicks it up, you will get a new one forming underneath. And so the direction of building here is actually to stick the chain of amino acids out the top of the ribosomal subunit. So the protein starts forming at the polypeptide site. Makes sense, right? It starts forming at the polypeptide site and sticking out of the large subunit of the ribosome. And this process of bringing in a tRNA into the A site, so the A site first, the polypeptide site second, and then the exit site doesn't actually hold on to the tRNA at all. It's actually a pair of scissors, a little tripwire. See if I can draw some tiny scissors. Oop, that's not the direction. Wait, is it? There we go. Look at those scissors. A little pair of scissors to cut the bond between your codon and anticodon, so that way the tRNA can leave and then go ahead and find a replacement amino acid. So that way the tRNA can be reused. So you'll have tRNAs entering the amino site, continuing on to the polypeptide site, where they get the previous amino acid stitched on top of them, and then being tripped up and exiting through the exit site, the third and final place of the ribosome. This is happening at an absolutely breakneck speed. And you've got this polypeptide or protein being built out the top end of your large subunit of your ribosome. The reason we call it a polypeptide is because this is what the functional top of an amino acid looks like. You've got your variants here. We'll take a look at amino acids next lecture. And so this variable R group always has these two groups, N's and C's, so NH2 on one side, and COOH, the COO group, carboxyl group on the other. And when you bind a amino or NH2 group with a carboxyl group directly to one another, we call that a peptide bond. Now notably, do you see the molecule that has to be removed in order to form a peptide bond? Well, it's got two H's and one O. That's right. Peptide bonds are formed through dehydration, synthesis. You need to remove a water from these two molecules in order to connect them. So the large subunit of the ribosome is actually producing a whole boatload of water the whole time it's assembling the protein. Now, the protein will be assembling out the top end of the polypeptide site, the P site, up until a stop codon is reached. Now, what is the difference between a tRNA that carries a stop codon and a tRNA that carries a amino acid? Well, a tRNA that carries a stop codon will not have an amino acid attached. It is carrying a blank. So when it binds to your A site and then takes over on your P site, and the previously elongated chain goes to try to stitch on top of it, there's nothing to hold on to. There's no amino acid to bind to. There's no peptide bond to be formed. So at that point, your amino acid chain just happens to float off. Now, I can't drag it particularly well because of graphical limitations, but your amino acid chain will float off and now form and be its own protein. As a refresher, protein structure happens in 4 different phases. I'll grab a thinner marker here. The first and primary structure of a protein is a straight chain of amino acids, like what you see assembled in the polypeptide site. But once enough of them are sticking out there, you will see that protein start to fold. And it folds into helixes, or alpha helixes, and beta pleated sheets. So that kind of looks like a corrugated roof, something that bends. Beta pleated sheet, or an alpha helix, kind of like a corkscrew. Those are your two different structures that define your secondary structure. Tertiary structure is when those alpha helixes and beta pleated sheets fold on each other to make a large 3D protein. Third structure, 3D. Fourth and final structure is when you have one 3D amino acid chain plus another 3D amino acid chain plus another three-dimensional amino acid chain. When you have many, at least two or more, 3D structures, 3 amino acid chains formed together, you form a quaternary structure of a protein. And we oftentimes refer to quaternary structure proteins by different names, like globins, because they are globs of many amino acid chains. You might be familiar with the globin called hemoglobin, because it requires 4 3D chains of the heme amino acids, alpha heme and beta hemes, to glob together to form one giant hemoglobin. So likely the protein that you hopefully are familiar with, because it's grabbing all of the oxygen that you're breathing right now, is actually a combination of 4 different amino acid chains that were each synthesized in separate translation reactions. And so that's where we'll end the lecture today. I do want y'all to spend some time modeling out translation, as you most certainly will be expected to draw this out in different stages. Note that I drew it out in one molecule to kind of illustrate how it looks and how it moves through time, but you can do this in iterations like a comic, so I'd encourage you to do so. Know what actually causes termination of translation, that being the tRNA that does not carry an amino acid that codes for the stop codon. What is causing elongation, the ticking along of different tRNAs in the A site, then the P site, then being tripwired out the E site, and the sliding along of the large and small subunits. And then what actually begins initiation, the binding with the 5' cap and UTR to the small subunit. And then know these processes for RNA processing, what the 5' cap does, what the 5' UTR, what each of the exons do, meaning that they are expressed, they make your chain of amino acids. And then know what the poly A tail does in a little bit more detail than everything else, because we spent some more time on it. And the 3' UTR, again, location afterwards. Know termination, elongation, and initiation, and the different promoters that we'll encounter in transcription. Know how to complement DNA in order to make the complementary strand of DNA, and then how to transcribe it into RNA, basically changing your T's into U's, right? So if you compare this sequence to this sequence, you'll see that this has T's, this has U's. That's your transcription. And then know how to read a codon table. Know your differences between your different RNAs, M's, T's, and R's. And that, folks, is the central dogma of biology. The central belief, the central observation, that one gene, one sequence of DNA, will be transcribed into an RNA that can then be translated into an amino acid sequence, a protein, that will go on and do stuff. Literally everything that we need to survive. Proteins are quite diverse. So on that very eloquent note of using stuff in a college lecture, I'll leave y'all to review your hopefully detailed notes and walk through the process of replication, transcription, and translation. So that way next time we can talk about control.